will also briefly introduce remodeling. That's a very important process. The chromatin remodeling, it's a very important process for a successful packing as well as unpacking. In both the level, it is very important. Because without these, you cannot have a successful opening or closing of the chromatin. Obviously, the underlying factor that contributed by the epigenetic modifications, whether it need to be closed or open, and then epigenetic modification play a very important role. So, why do cells require the chromatin remodeling? Right. So, because when a cell want to express or particular gene to need to be expressed in the cell to mRNA or transcribe, the DNA is not freely available, right? So, DNA is a bond with histone proteins. That means the DNA need to be accessible to the transcription factors to initiate the mRNA synthesis. So, for that you need to really move these histone proteins to make a space for the transcriptions. So, for a successful transcription, the DNA should be freely available. That means histone need to be moved further or slightly slided on the DNA structures for a successful accessible for the transcription factors, right? And the packaging of the DNA is very important because it is generally DNA histone packaging. Although it is the charge favors this, it's not. It's that easy process. When you do it up, it just randomly assemble. But in a cell, the proper packaging is very important. In the in vitro, when you have the DNA structure and histone, when you mix it up, yes, it may assemble randomly, but the proper assembly is very important. For the proper assembly, you need to have a factors or proteins. The nucleosome remodeling is very important for the successful packing. So, for all these things, the cell system evolved with the specific mechanisms, the remodeling. So, in large, this is a very big topic. I will introduce the basic line that is enough for epigenetic context, so that you will have a knowledge. Yes, this is how the nucleosomes being mobilized or moved further for a successful opening or closing of the chromatin. So, in connection with the remodeling, it will does at two level. Simply by using the ATP dependent manner, that is ATP dependent chromatin remodeling. Most of the chromatin remodeling actually takes care of it. There is a group of protein which comes and contribute for this process actually. So, mostly what does it do? Mostly doesn't remove actually the histone proteins out of the nucleosomes. So, it will have a nucleosomes packing. So, basically it helps to move the nucleosomes or histone proteins further, mobilizes, and then transcription factor binds here and helps to produces. Then later, after polymerase moves further, and then this will be reassembled. So, basically for that purpose, this nucleosome remodeling is essential. The second important place where it contributes, packing. As I always say, the packing from these euchromatin to heterochromatin, nucleosome remodeling contributes along with the epigenetic modifications. I will give the overview about the types of chromatin remodeling. Of course, it's a very big chapter, but I'll just make it a more general overview, so that you'll get a logic of how chromatin gets compacted, and then which is essential for the chromosome shaping as well as gene expression and so on. There are two types of chromatin remodeling. The histone covalent modifications. So, basically that is an epigenetic modification driven chromatin remodeling. Obviously, all the remodelers which I am going to tell in a subsequent slides, which is rely on this, but sometimes independent of these second types, that is ATP dependent remodeling, this alone takes care of compactions and sometimes relaxations. So, that is why this is classified separately, the epigenetic modification dependent. This is without this in remodeler, this can happen, the histone modification dependent chromatin remodeling. The second one, ATP dependent chromatin remodeling, it is not always require the epigenetic modifications, but there is a strong interplay between these this one and then epigenetic modifications. So, I will highlight these ATP dependent chromatin remodeling structures. Obviously, we are going to discuss in detail about these various histone modifications and its role in gene expressions. Right. So, the ATP dependent chromatin remodeling, uh, it's basically a group of proteins. It's a complex which primarily involves in the either gene expressions or chromatin accessibility, mostly in connection with the gene activation process, where it assists in moving, ejecting, or restructuring the nucleosomes. Because the DNA is wrapped on the histone proteins, DNA need to be accessible for these gene expressions. So, for this purpose, the group of proteins which uses ATP as a energy, and then which does these kind of functions, either moves the nucleosome on the DNA or removes, eject, or even restructure. That means it can add a new isoform of the histones proteins and then remove the old one. There is a common between all these ATP dependent remodelers. All has the ATPase domain, which uses energy by hydrolyzing the ATP. There are four major family comes under this chromatin remodeling complex. One is a SWI SNF family, it's a large family, and then ISW high family, and then CHD family, and then INO 80 family. These are four major family. So, among these family only, all these node complex, nucleosome remodeling complexes and all will comes under this. Okay, this is a classification of these chromatin remodeling complexes. All are multi-protein complexes. It's not a single protein. There are group of proteins involved which assist in chromatin remodeling. Majorly, as I said, to help in the relaxations or remodel the chromatin structures, which is more accessible for the gene activations. This one, the CHD family, is the one which is little bit exceptions, which is involved in the chromatin compactions or gene repressions, which I will highlight. Right. So, as I said, all are multi-protein complexes. All the chromatin remodeling complexes, not a single. And then in that there is a catalytic subunit, which is mostly ATPase domain, which uses ATP as a energy source to conduct either ejection or sliding or removing of these or restructuring of the histones protein on the nucleosome structures. And there is a all has the common ATPase domains, right? But there are some differences in the protein which conducts the which carry out the remodeling, which is specific domain which is present. See, for example, SFI2 or SNF2 members largely contains the bromo domain. It is nothing but is a protein regions within the protein, which has a specialized module. It has an ability to bind to the acetylated lysines, mostly the bromo domain, which we will see in a detail. And then ISW high member, which has a SAN domain. This is also the epigenetic reader family. So, this is also epigenetic reader, this is also epigenetic reader, which all binds to the modified histone proteins. Whereas the CHD member, it has the chromo domain and DNA binding domain. And then another family, the fourth family, INO 80, which does not have any of these domains, but however the ATPase domain are splitted using these linker regions. So, these are the key differences between these four families. I will highlight the key important function about this each family. Coming to the SWI2, this is actually named switch defective or sucrose non-fermenting SNF. Basically, it was identified from the yeast Saccharomyces cerevisiae. When they try to screen the phenotypes with the various mutations, they found this family shows these kind of specific phenotypes. That's why the name was given. It nearly has 8 to 14 subunits. It's not a single protein, as I said, it's a multi-protein complexes. And this family highly conserved across these two mammals. So, what is the key functions of this family SWI2 SNF, which is primarily involved in a sliding of the nucleosomes, basically stones. Sliding means you have this uh, uh, DNA structure, I mean nucleosomes, the nucleosome will be rolled on here. So DNA will be free. So this is the sliding mechanism. So it will be nucleosome will rolling and DNA molecule will be free and eject. As I said, the subtypes or histone isomers, one of the isoforms can be removed and then new form will be added. So these family primarily takes care of it, mostly involved in the gene activations. So these 5-2 SNF family chromatin remodeler, that means helps to relax the condensed chromatin to the euchromatin basically, or wherever the specific gene need to be activated, this will come and help for the gene transcriptions. The second family ISW high, that is imitation switch remodeler. It is relatively small number of proteins. You see the other one is nearly more than 10 proteins as a complex, but ISW high is only 2 to 4 subunits, sometimes even 1 or 2 in some organisms, not many subunits required for this. And this also involved in the sliding and ejections. And this is also involved in the gene activations, because it has the bromodomains. Bromodomain is known to bind with the acetylated lysines. Acetylated lysines on these stones are gene activation mark. So through these bromodomains, this will bind to these and helps for these gene activations and remodels the nucleosomes. So basically, the second family also linked with the gene activations. So that means it is also performs similar function like the first one. The third family, it is a chromodomain helicase DNA binding. It was identified as a part of these Mi2 remodeler complexes in the xenophus, and sometimes 1 to 10 subunits. The mostly these also involved in the sliding of the nucleosomes or eject the nucleosomes to promote the gene transcription. Again, this is a context specific, and some it will be transcription and some will be gene repressions. So that means it depends on the association of the complexes, because the complex matters a lot. I said 1 to 10, this is a dynamics. That means in some positions can be 6 proteins, some position will be 7 proteins. So the complex nature, which are the protein associated along with the ATPase domain, which decides the whether gene to be activated or gene to be repressed. So that's a context dependent, that's the meaning. So yeah, so it participates both in a gene activation as well as the gene transcriptions. The fourth family, INO80, that is the inositol requiring 80. So this is based on again gene knockout studies in the Saccharomyces cerevisiae, which also carries nearly 10 subunits. These are all multi-protein complex, all the family. The unique feature, as I said, the ATPase domain of these remodeling complexes are split with the with the middle insertion sequences, which separate the ATPase domain. That's a very unique compared to the other 3 family. Other 3 family is single ATPase domain. Within this is the only one family. The ATPase domain is splitted like this. So insertion sequences and then split, but it still performs the ATPase activity or ATP hydrolysis for the chromatin remodeling. It primarily involved in the gene activations and DNA repair. See, this is the only family mostly involved in the DNA repair mechanisms along with the gene activation. Gene activation by default you can imagine or you can think about it. Yes, the sliding of nucleosomes on the DNA and ejection as well. But in addition to that, INO family conducts the DNA repair mechanism as well. Particularly, it does 2 level. One is H2A point Z. I said, do you remember I said H2A point Z is for a variant for gene activation. So with histone variant replacement. So directly it helps to insert this histone 2A point Z variant and which is a marker for gene activations. Whereas the another variant H2A point, sorry, H2A point X. So this is the DNA repair histone variant. So this has a specific phosphorylation site. I said serine 130, which get phosphorylated, and then which marker for the DNA repair. So then it recruits the DNA repair machinery. So basically, this remodeler, along with histone chaperone, it also helps to replace these histone variants in the specific loci. In addition to this H2AZ as well as H2AZ. Right. So this is the key features about the all four family what I explained. For information, as I said, this is very diverse topic. So I am not going to explain about each topic's details. I want to just give a glimpse so that you will aware about it before we start discuss about the histone modification dependent gene activation or repressions. So that you familiarize, it's not only the histone modification involved in the chromatin compaction or relaxation. There are other proteins which participates. These proteins may require histone modification, sometimes may not require. But yeah, majority cases it depends on the chromatin modification dependent recruitment and then activation or compactions. That means all these protein has a specific reader module. I said bromodomain, sand domain, chromodomain. So these are the reader modules which recognize the various modification on these stones and then get recruited to the specific loci. So the common features about all the chromatin remodeling complex, they do bind to the nucleosomes, and then they are DNA dependent ATPases, all as the ATPase domain. Of course, as I said, all are recognizing various histone modifications. The histone modifications on the tail is an important docking site, which we will see in all upcoming classes how it contributes for the gene expression. I am going to explain gene expression activation, but underlying you should also keep in mind either relaxation of the heterochromatin or compaction of euchromatin to the heterochromatin. The modification also contributes actually by recruiting various chromatin remodeling complexes. Right. So the ATPase activity, as I said, is a multi-protein complexes. The interacting protein also contributes in regulation of the ATPase activity. So these are some sad features of the almost all the chromatin remodeling complexes. So mechanistically, it slides the nucleosome on the DNA structure, which makes a space for to bind to the RNA polymerase or transcription factors. And in specific cases, it can eject the histone octamers. That means it can remove and then add a new histones. So this is one typical phenomenon. But there is a recent discovery that's called you have these histone, so nucleosome structures. Then you have 8 tails, right? So in the specific nucleosome regions, what happens? The cell undergoes for the removal of these tail histone tail truncation process. So what I said, ejection, it's very common. Ejection means it just removes the kind of repressed nucleosome. I mean, histone, one of the histone subtypes adds a new one, which may be helpful for the DNA repair, activation, repressions. But very few cases it has been identified. It doesn't do with the any histone isoform ejection or removal. Rather, it cuts the tail. When you cut out the tail, see all the complex mechanisms will be prevented. See, the tail is the one important place where the modification added, and then reader protein has to come and then mediate all the things. So the specific place just cuts out and then free to access to the DNA structures. So this is very small function and localized regions, not a global. Yes, but cell also does like this. That is information you have to keep in mind. Yes, also it removes these H2A, H2B dimers and replace the new variant which I already explained. And then all the remodeling complexes, it has an ability to bind to the DNA, of course, because DNA protein complexes for a successful any one of these mechanisms. Right, I'll just show you schematically how this sliding happens. I explained sliding in almost all the chromatin remodeling. So how exactly the sliding happens? You can see here, this is the nucleosome array, arranged structures, and then either it can move the one of the nucleosomes here, so make a space for the uh, uh, transcription factor binding or RNA polymerase, or it can just simply eject this one or make a more distal space. You can see there are a lot of space in each. So basically creating a spacing. So this is due to the sliding. You slide the positioning of the nucleosomes. It's a very important mechanism. Nucleosome sliding is one of the key mechanisms for the chromatin remodeling. So this is how it does. This almost all the 4 remodeling complexes what I explained are participate in this process. Yes, ejection, that is a removal. You can just see that the unwanted nucleosomes can be removed, and sometime it will be replaced. Okay, so after the transcription is done, and then once the transcription activity done, then this can be added and then repacked and then maintained inactive. So that is we consider as the removal of the histone from this DNA. The replace, okay, and the removal, I said it's just removal and then it will be empty DNA structures. In this case, particularly in connection with the H2A point X or any other histone variant A point Z. So X, keep in mind always it is a repair. H2AX means it's a DNA repair marker. So wherever the DNA repair happens, this histone variant immediately replaced, and this variant Z is a gene activations. It will be replaced completely. So these are also chromatin remodeling mechanisms through ATP dependent process. Right. So I schematically explained, right, but experimentally how people they concluded. So these are all theory initially, the hypothesis, but always you should have a solid experimental data to to confirm yes the complexes does sliding, right. So how they reach out actually does sliding. So this is one of the beautiful biochemistry assays they conducted in early 2000 to show yes chromatin remodeling complexes indeed can slide the nucleosome positioning when they conducted at a single molecule level. So they used the chromatin remodeling complex called ACF, ATP dependent chromatin assembly factor. It's one of the large subunit. What they had done, this is a part of this ISWI containing, okay, the ACF is part of this family. So they did a nucleosome remodeling assay with the single you have this nucleosome structure you can see, okay. The position is at the end now. So after incubation with these complex ATP dependent ATP on all the substrate buffer the protein complexes, we see this is a time dependent, this is a time increasing time. They see the mobility shift. In the beginning you see the mobility is down because of the positioning of the nucleosome here, right, and over the time if you see the shift from here to here, you see this shift, that is due to the nucleosome position in the middle. So the positioning of the nucleosomes decided or decides the mobility actually on the gel. This is a basically 4% native polyacrylate gel, very tiny gel. So what is a normal gel? We use it for electrophoresis, DNA electrophoresis, page, native gel maximum 8% is the best you can handle it. Okay, I'm not talking about SGS page. This is the a DNA gel for a DNA page gel. So 8% the best, but you see they use really low percentage, 4%, very difficult to handle. Such a low tiny gel, low high porosity but low concentrations, because that allows for the proper separations. You see the very strong biochemistry evidence. They conduct time dependent manner and then they use this is 0 hour 30 minutes to long time, and each time points they loaded and then they see time point dependent shift in the presence of ACF, which clearly shows that indeed the nucleosomes are sliding. That is why you see the shift, right. So this is an experimental data. Whatever the theory, you can always make logical conclusions, but you will make based on these strong experimental evidences, strong biochemical data, where in that study they reported beautifully, which I highlight in upcoming class as well. Histone 4 tail, okay, not histone 3, histone 4 tail, and particularly amino acid 16 to 19 is essential for these remodeling complexes. I'll tell you why. Okay. So, yeah, but the bottom point you have to keep in mind, when they conduct a nucleosome remodeling assay with nucleosome, it does slides and then it moves inside, I mean moves upward, and then that's due to the sliding of the nucleosome on the DNA structures, right. So, much more detail, I explained this structure, the Histone 4 tail. Let's imagine this is the nucleosome structures, and then I will show you the structure later, then you will connect it, but I will try to introduce right away. There is another nucleosome structures, right, and then there is a Histone 4 tail within the nucleosome itself, in this case single nucleosomes, H4 tail, which basically interact with H2 proteins, H2A proteins. H4 tail binds to the H2A, H2B. So, this interaction is essential for the successful association of the single nucleosome. Got the point? So, the tail of this H4 and H4, you will understand when I am showing the structure in upcoming acetylation class slides. Now, I will introduce in connection with this assay. Okay, so the tail is essential. So, the tail, positively charged tail. Okay, so that's why these regions are positively charged regions, and that is associated with this H2H2B negatively charged patch on the H2H2B, which helps to retain the histone proteins complex. What happens when these regions are acetylated, and because of charge imbalance, right, this will get disrupted. This was a discovery from that paper. Here you see, if don't think about this one right then, we just look at this as a wild type chromatin remodeling, time dependent, till 60 minutes they conducted, and then they see the good shift. So, most of these nucleosomes are shifted up, right, it is very clear, but here there is a delay. So, the nucleosome remodeling is delay because then there is a acetylation at these positions. So, they are trying to say the acetylations prevents the remodeling of this ACF. How exactly mechanistically, I will tell you later. Okay, so bottom point from couple of slides, the whole idea I want to explain, the chromatin remodeling experimentally they confirmed using this particular protein, ATP dependent chromatin assembly factor. So, it does when they mix this protein complex ATP and so on with a single nucleosome structure, it does shift. That is because of the sliding, and mechanistically, that is why it is science paper in 2006 itself, very strong paper. They went ahead and showed it, the acetylation of these particular positions, okay, 16th positions of histone 4 protein play very important role in disrupting this H4 versus H2H2B, and then that gets disrupted again ACF. So, there is a complex mechanisms. When I show a structure in upcoming class, you will understand how does it works. Right now, you keep in mind the histone 4 lysine acetylations prevents the ACP dependent chromatin remodeling, but this is also gene activations. That means this ACP may involve in the chromatin compactions, right? Good. Right. So, this is other one of the examples, nucleosome remodeling factor, NUF, which has the specific domain which bind to the H3K4Me3, which is a strong gene activation mark, only known gene activation methylation mark on the histone proteins, histone 4 lysine, histone 3 lysine 4. So, this remodeling complexes binds to this particular histone 4, you can see here, and then which helps for the remodeling or gene transcription. So, this is how the K4 modification function as a gene activation mark. So, we will have a detailed discussion about K4 modification, and this is one of the mechanisms. See, mark will be added, epigenetic mark. So that alone doesn't mediate. Acetylation makes sense because it brings a charge imbalance, but methylation does not bring any charge differences. It always recruits the molecules or protein complexes. It functions as a docking sites, and then it brings either repressor or activator proteins. So in this case, activation mark, how does it function as activation? It brings one of the important remodeling complexes, nerve2, which helps the opening of the chromatin and then facilitate for the gene activations. Right. So this is a model to, you can imagine how during remodeling process, particularly gene transcriptions, because we talk about gene transcription, we never imagine how exactly the polymerase travels on the DNA structures, because DNA is wrapped on the histone proteins, right? If you imagine how exactly it travels, this is how it travels. So of course these are all which depends on the sliding mechanisms. You can see the polymerase enzyme transcription machinery which moves on these DNA structures here, and then while moving on the histone core, this is the core regions, and then it doesn't completely remove all the DNA structures, right? It just makes a space where the enzyme can travel and then produce mRNA in this case, and then it wraps up. So this is how most of the transcription takes place, gene expression takes place by coordinating with this chromatin, nucleosome sliding, slightly move around and then make a space for the enzyme to move forward, produce the mRNA. So this is the, so far I gave an outline for the chromatin remodeling largely in connection with the sliding ejections with all the family, and then I briefly introduced the experimental evidences, how people have confirmed the sliding happens. Of course, later after that's a classical biochemistry assays. Later, people have used the chromatin immunoprecipitation assays to show the replacement of the histone variants on a specific loci. That's much more powerful assays came inside, and where all these assays were confirmed to show yes, replacement happens, ejection happens, and then sliding. So all these things has been done with much later. Okay, I mean now this attack sequence came up, so there are a lot of sequencing technology came up to show the chromatin compaction, relaxation, and a specific loci. And so all these experimental evidences came up much earlier, but these are all the data people understood in the early days itself using the simple biochemistry assays. Okay, so now let's move on to an important topic. So for the past 2 or 3 classes, we have discussed in general chromatin structures and packing, how does it pack, and then the role of chromatin remodeling machinery. And now I will move on to an important topic, histone tail and core modifications. So the histone epigenetics, it's a huge topics where I'm going to highlight only the tail and core modifications. So what is tail and core? Just to recollect again, you have a nucleosome structures, right? This single nucleosomes, this is the DNA, linker DNA, and then this is the tail, histone tail. This is H3. It is the largest histone tail. I said this nearly 1, 2. This is the N-terminal tail. So all the proteins N-terminal are unstructured, which I already explained, which is projected out. And then H3 is nearly amino acid 1, 2, 37 or 38. It's a pretty long, right? And H4, second largest, which is 1, 2, roughly 24 to 25. So this many length amino acids does not have any secondary structures. So this is how the tail function in the cell. When in our nucleus, in our cell within the nucleus, the tail are unstructured. But this is the tail regions. This is histone core or globular. So you can call histone core or histone globular domain. So both the term you will find in the textbooks or literatures. So the whatever the modifications present on the tail, you will call histone tail modifications, and its associated function activation repressions, and there are modifications happen in the core. When I explain the core modification, you will understand how this functions, how the core modification functions. Each function differently, but largely either chromatin compaction, relaxation, or activation repressions. It has either gene activations. If you think of superficially, it has to do either repression or activation, or relaxation or compactions, right? But mechanistically differ. The tail modification does different mechanisms or execute through different mechanisms, and this execute through different mechanisms. So this is, so I will use the core modification in almost upcoming classes. That means you have to imagine the core modification means modifications present in the core region of nucleosome. This is the core regions. It can be H2A, H2B, H3, H4, any of the histone proteins. And similarly, tail, almost all the tail modifications, but the major epigenetic modifications which we will see on histone 3 and histone 4. Recent evidences and many histone as well people have reported, but the classical epigenetic modification you define as a canonical or classical, which is well studied, which is linked with the inheritance and epigenetic phenomena. So that alone I will explain in detail in upcoming few classes. Right. So you see there are many positions gets modified. It can be, it's in a H2A or H2B. The most abundant modifications and H3A. There are a lot of residues gets modified, and H4 as well. So what are the key modifications happening on these histone tail proteins? Core we will talk later after completion of the tail. Okay. The major post-translational modifications which is happening or occurring on these histone proteins, methylation is a very important epigenetic modification. You see the structure, small histone, so small methyl groups, tiny methyl groups. And then second important is acetylations. It's slightly bulky, right? The methyl group is tiny structures, chemical structures, and this is bulky structures. And phosphorylations, phosphate group is added to the serine or threonine. And ubiquitin or smolation. You see these two are pretty much big structures, either ubiquitin or smolations. Both are happening on the histone proteins. So when a specific residues, I am not going to explain in detail in epigenetic class either about smolations or ubiquitinations or phosphorylations, because I am going to concentrate only methylation mark on acetylation mark extensively and its a role, but phosphorylations and a context specific I will introduce very briefly, not in detail. And ubiquitination smolations I am not going to explain because it does not have much of epigenetic functions, so which is not relevant to our epigenetic class as well. Right. So all the modifications are covalently attached, that you should keep in mind. It's not any other simple bonding. Attachment is to them because methylation acetylations happening, methylation happening on the, or methylation takes place on the lysine amino acid, then arginine. These are two important residues. In addition, glutamine as well. Okay, so these are recent studies identified. Yes, glutamine also gets methylated at some extent. But of course, its function is still undergoing with investigations. But the major modifications are linked with the gene activation or repression, chromatin remodeling, and so on. Lysine acetylation, lysine methylations, arginine methylation. Similarly, acetylations only on lysine. And phosphorylations, you all know serine, threonine. Right. So all these modifications, methylations or acetylations or phosphorylations are dynamic. That means it can be added through by enzyme or reversible. So that is why you define as a dynamic. So both additions of the mark and then removal done by the enzymes. There's a group of enzymes involved in this process. Why we need a dynamics? Because each cell has a different phenotypes or identity, different expressions. So to have a successful gene functions, the dynamic activity is very important. Having dynamic epigenetic signature, that is why you have the cell identity, right? So for that, the dynamicity is very, very important. and of course each modification has a diverse biological functions. To make it much more clear for you, I already said methylation mark is very tiny, it's a small, and then acetylation is large, and you see it's a very tiny one in methylation acid, but the ubiquitin molecule size is almost 8 kDa, or protein size, histone proteins around 14. So histone 3 maximum 16 to 17 kDa, histone 4 it is, there is some error here, but mostly 11 kDa, okay, and then H2A it is 14 kDa, and H2B it is around 13 kDa. Okay, so when you extract histone, when you run a gel, you will have a pattern like this. That pattern you should at least keep in mind. So when you run a gel, you have this histone 3, H2A, H2B, and H4. H4 is the smallest protein. H3, H2A, H2B, and H4. So this is how you will have the protein band separation on the SGS page. Okay. One of the unique features about these methylation mark, it's a tiny one, but when it is added onto these lysine amino acids, there are 3 functional groups. So the enzyme can introduce a 3 methylation group. So that is why you name as a methylation 1 or Me1, Me2, Me3 in the textbook. The reason the lysine, in this case it named as a histone 3 lysine 4 Me1. That means the lysine has the monomethylations. That is because only one methyl group added here. And Me2, if we define, it's a dimethylation added, and Me3 it is a trimethylation. It is a sequential. So most of the cases single enzyme can perform. So an enzyme can convert lysine to monomethylated lysine. So this become a substrate for the next reactions. So that the mono become di, a di become a substrate for the next reaction. So tri. So the 3 methylation groups very unique. So 3 methyl groups are added. All 3 are, when it is 3 methyl group added, that is the functional mark. What is functional mark? Means it can execute whatever the function, repression, activation. So basically the complete methylations, in this case trimethylation mark, involved in the successful functions. But I will also highlight in upcoming class, there are examples, the dimethylation on a specific nucleosomes will have a very specific functions, and monomethylation on some nucleosome will have very specific functions. So these are exemptions. Either zero methylations that you call Me0 or Me3. So this has the defined functions, either activation or repressions. Whereas the intermediate mark also do present on a specific gene sets, but that has very specific functions, and in specific cases I'll highlight. So this is how the naming was given. So this is how you should write always. You should not write in it, because this is the accepted way of writing to write about epigenetic modifications. Any modification, talk about it on any proteins, you should always write either histone 3, subunit of histone 3, and then which lysine, and the 27 means 27. And if this position can be acetylated, you should write acetylated, right? Our histone 3 lysine 27 methylated means you should write Me1 or Me2, whatever you identified. So this is how you define it. Right, come. All the marks, acetylation or methylation mark, or has this specific epigenetic modules. So all the marks are added by the enzymes that you define as a writer. So enzymes, it can be methyl transferases or acetyl transferases, which adds or writes the methylation mark or acetylation mark on a histone tails. So that you call as a writer, which adds the mark, and a reader, which reads or dictates or brings the function for the modifications. So of course the acetylation mark, as I always said in multiple classes, which directly can influence the structure of the nucleosome by bringing the charge imbalance. But methylation mark doesn't do anything. That means it will become a docking site. So then there are other protein can bind to these modifications and then mediate a functions. So the classical example just I explained, histone 3, lysine 4, Me3, where the nerve complex, right, binds and mediates a chromatin remodeling dependent gene transcriptions. So that you define as a reader. Bromodomain, chromodomain, pH domain, these are all epigenetic reader module. So what does it do? It goes and binds to the modification, and then this became a docking site for the either activator or repressor complexes comes and binds. So basically that interpret the function, whether to be activated or repressed that particular nucleosome, or the nucleosome need to be compact or relaxed. Coming to the eraser. So this is called as an epigenetic writer or a reader, an epigenetic eraser. Eraser is an enzyme which removes either demethylase or deacetylase. Since I said these marks are dynamic, wherever it need to be added, it will be added by enzymes, and where it does not require and then can be removed. So where this eraser can come inside and take care of to remove. It's a very important process. The eraser is play very important role, and this dynamic is play very important role during stem cell differentiations, early stage of development. Same like DNA methylation, what I explained, erase DNA methylation is removed and then added, and then similarly when the cell committed to the specific lineages, it has to switch on or switch off on specific gene, right? Tissue specific genes to be switched on, or some genes not required to be switched off. So the switching on, switching off during these lineage differentiations to be done. That means some mark to be removed, some mark to be added. So that means the dynamics are highly essential for a successful development. So that's a place the eraser comes and remove. Where it does not require, goes and demethylate or removes it. Okay, so you see here acetylases, methylases or phosphorolases all writer, and then all deacetylases, demethylases or phosphatases or eraser, and bromodomain, chromodomain, PhD protein domains, all are the readers. Right. So which already explained, the lysines undergoes for the acetylations, lysine undergo for methylations, lysine for ubiquitinations. So we will have a detailed discussion about these enzymes. You will understand, because similar to the DNA classes, DNA methylation classes, you should have a knowledge about these enzyme and reader as well. That is why I introduced this topic, because by knowing histone 3 lysine 4 methylation mark gene activation, it should not end up by learning only epigenetic. You should know who does it, how this function, how this mark functions. So who does it mean? You should know who is a methyltransferase. How it function, you should know the reader, right? Then only you will understand the mechanism of the epigenetics. So by just memorizing our histone 3 lysine 4 gene activation mark doesn't help you much. It will keep you something like one line answer. But to know, understand the depth of epigenetics, you should know who does it. So that's why I'm going to cover up in upcoming classes the enzymes, along with I will talk about the modification function and then enzymes. So we like structurally we'll go for the modifications, and then writer, and then a reader, and then eraser, key modifications. Then in all upcoming classes we keep discuss about these directions. Okay.